So the first one is a paper published in a journal called European Surveillance, January 2020. The author is a guy named Cowan and others. And the title is Detection of a 2019 Novel Coronavirus by Real-Time RT-PCR. So this is a paper in a peer-reviewed European journal about detecting this coronavirus. So here's a quote. Quote, "The ongoing outbreak of the recently emerged novel coronavirus, 2019 nCoV-2, poses a challenge to public health laboratories as virus isolates are unavailable." So what does that mean? It means that it turns out these virologists, I think there was about 20 of them, were tasked with the job of developing a test for finding this coronavirus. But as they said, this is a challenge, and I would admit that that is one part of that sentence I actually agree with, because it's a challenge because they don't have any isolates of the virus available. In other words, they don't have the caffeine. They don't have an example of the virus. And it would be very challenging to make a test to find something which you've never seen. So I would agree with them on that point. The analogy I made, it's like you're told to look for unicorns, and you can look for hoofs and tails and hairs and things, but you're not allowed to actually see a unicorn. And it could be because unicorns don't exist. So then they go on in the study and say, quote, "We aim to develop and deploy robust diagnostic methodology for use in public health laboratory settings, without having virus material available." And as I said, I had to read that a number of times because their job, what they're saying, is to develop a test that the test is based on finding a unique piece of this virus, knowing that this unique sequence could have only come from that virus. Therefore, they can look for the sequence instead of the virus. Yet, as they admit, and this is January 2020, they don't have any virus material available. And I can imagine how challenging that would be. So I'll leave that one for a minute. The next one, people have heard me say this. This is from a more recent study on the CDC document. So this is published by the CDC itself. The title is called Novel Coronavirus 2019 nCoV-2, Real-Time RT-PCR Diagnostic Panel, published July 13th, 2020. On page 39 of this long document, we read the following, and I'll quote, "Since no quantified virus isolates of the 2019 nCoV-2 virus are currently available, assays, parentheses, diagnostic tests, designed for detection of the 2019 nCoV RNA were tested with characterized stocks of in vitro transcribed full-length RNA." Again, in other words, this is a document describing in July 2020, so this is well into this pandemic, of how they go about developing these PCR tests. And they say, quote, "There are no virus isolates available." In other words, they've never actually found the virus. They've never found the caffeine. They've never gone through those steps. Therefore, you don't know whether there's even caffeine in there. And so instead, they use their testing sequences that are based on stocks of in vitro, that means laboratory created transcribed full-length RNA. In other words, back someday, they made these full-length sequences in the Petri dish, not from isolating them from anybody or anything. And that's what they're using as the blueprint, the template, to see whether they actually can find the virus. So that's crazy. The third is a paper that was published in June of 2020, also by the CDC, in CDC Bulletin Volume 26, number 6. The author of the paper is Jay Harcourt and others, and the title is Severe Acute Respiratory Syndrome Coronavirus 2 from Patient with Coronavirus Disease. And let me read you the quote from one of the sections in this. The authors state, quote, "Therefore, we examined the capacity of SARS-CoV-2 to infect and replicate in several common primate and human cell lines, including human adenocarcinoma cells, parentheses A549, human liver cells, parentheses HUH 7.0, and human embryonic kidney cells, parentheses HEK-293T. Each cell line was inoculated at high multiplicity of infection and examined 24 hours post-infection. No CPE was observed in any of the cell lines except in vero cells." Now, that's a big, long mouthful, and I don't expect anybody to understand what they mean or see the incredible significance of that paragraph. So let me dissect that a little bit. First of all, CPE means cytopathic effect. So here's why that is significant. So if a virologist wants to prove that a certain virus, isolated or not, causes disease, they have 3 different possible hosts in order to test it on. The first host obviously would be humans, people. The second host would be animals. And the third host would be tissue culture. In other words, tissue taken from a kidney tissue of a monkey, or human tissue from the lungs, or human fetal tissue, or eggs, or whatever other living tissue, because obviously we all know viruses, quote, don't grow in petri dishes or on non-living medium. So the question is, have we proven, because that's the point of this, is to demonstrate that this coronavirus causes disease. So there are obviously no examples where they've tested this isolated or not coronavirus on humans, because clearly that would be unethical, right? So we have no examples of this. So we have no direct knowledge that this coronavirus causes disease in people. So the reason for that, they say, is because that would be unethical. Now, what about with animals? So as we know, and we dissected in the book, there's essentially 2 studies that have attempted to use unpurified, unisolated virus fluid. That would be like the coffee. So they injected the coffee, which has a lot of things, into a bunch of wild mice, and none got sick. And then they injected them into genetically engineered mice to have fur problems when stressed, and a statistically insignificant number of them, in fact, got fur problems when injected with this unpurified stuff, which supposedly contained the virus in it. And so essentially they demonstrated that they couldn't make normal animals sick with the virus or a fluid supposedly containing the virus. And even animals, i.e. mice, programmed to get sick, they could only create a statistically insignificant number of them got sick in a way that had nothing to do with what's called COVID-19. In other words, they have no evidence that this virus or this fluid containing the virus causes disease in animals. So the next step is you have to do it on tissue culture. And in fact, most virological studies are not done on humans for supposedly ethical reasons. They're actually even anymore not done on animals because they say there's, quote, no animal models for this disease, which is an interesting thing to say. What the translation is that it doesn't make animals sick. So most of the proof that the virus causes disease, or more technically, the fluid which supposedly has the virus in it, is by inoculating this fluid containing the virus onto various tissue cultures from various sources. And so that's what they did. And shockingly, what they report, and by the way, we all know that if you just inoculate virus on tissue culture, nothing happens. We know that for 50 years. So what they have to do in order to make the, quote, virus have a cytopathic effect is first starve the tissue. They do that with what's called minimal nutrient medium, which means they take away the nutrients from the tissue. And then they add usually gentamicin and amphotericin, which are two known nephrotoxic drugs, to a tissue culture of kidney cells. Nephrotoxic means toxic to the kidney. And of course, just those alone, starving and poisoning the tissue culture, causes the tissue to break down in its constituent parts, which can contain things which we now know are exosomes, which are way tissues detoxify themselves. But anyways, let's forget about that particular criticism of the whole phenomena of proving tissue culture. Let's see what they found. What they found was they tested their unpurified liquid that supposedly contains virus on 3 different types of human tissue: human lung cancer cells, normal human liver cells, and human embryonic kidney cells. And they said they inoculated them at high multiplicity, which is a technical way of saying they put a lot of stuff on there, right? They put a lot of virus in there. And then they looked at them 24 hours later, and no CPE was observed in any of these cell lines. In other words, the only conclusion that any sane or rational person could come to is that the very way that virologists prove that this fluid containing virus infects and causes damage to human cells or human tissues, what they found was it doesn't. And they make no comment on that except to tell other virologists that, by the way, if you want to grow more virus, don't bother using human cell lines because the virus doesn't grow in human tissue or cell lines. And when you read that, again, the whole thing is sort of shocking, because if you just spent using your own technology, using your own methodology, you just actually proved that fluid containing this virus does not affect human tissue. In fact, they said it only affects vero cells, which are monkey kidney cells, which are sensitive to amphotericin and gentamicin. And that's the only way they could prove infection. The final one is shocking for a different reason. See if I can find it here. Hang on a minute. Yeah, sorry. So this was a paper published in the Journal of Clinical Microbiology. So probably one of the most important microbiology journals in the world. I can't pronounce the Chinese guy's name, Yu Zhang something, W. And others, the paper is called RNase resistant virus-like particles containing long chimeric RNA sequences produced by two plasmid co-expression system. That's a mouthful. Journal of Clinical Microbiology. So the point of this study was they were looking at novel ways of characterizing viruses. And so I'm going to read the quote from the study. This is under figure 3 of the study. And again, it's going to sound like gobbledygook, and I'll try to translate it as best I can in a minute. And here's a quote. Lane 1, positive control of PET MS2-3V plasmid using the primers S SARS-1 and HA300RT-A. Lane 2, RT-PCR of SARS-CoV-1 plus SARS-CoV-2 plus SARS-CoV-3 plus HCV plus HA300. Now, what does that mean? What they're saying is they developed new techniques, maybe even a little different, maybe even a little better than the usual PCR. So they were innovative virologists. And what they found was they could characterize different viruses using different primers. These are sequences. And so they were reporting on their characterization of the different SARS viruses. And so they were characterizing SARS-CoV-1, SARS-CoV-2, and SARS-CoV-3. Now, many of us, including myself, actually didn't know there was a SARS-CoV-3. In fact, we just have information that the SARS-CoV-2 was recently discovered because it had never existed before 2019. And there is no report that I know of that there's now a new SARS-CoV virus called SARS-CoV-3. But yet they're using their novel two plasma co-expression system, that's what the title says, to characterize this SARS-CoV-3 virus and actually put a picture of it. And one can only ask, what in the heck are they talking about? Because nobody, as far as I know, thought there is a SARS-CoV-3 virus. But here's the really shocking part of this paper. I could say, anybody want to guess when this paper was published? My guess is most people would say last week, last month, maybe at the end of 2019. The reference is Journal of Clinical Microbiology, 2007. Wait a minute. So 2007, they were characterizing with this method SARS-CoV-2 and SARS-CoV-3? I mean, supposedly it wasn't invented. I mean, sorry, I didn't mean invented. I mean, imagined. Sorry, not imagined. I mean, it didn't mutate into this virus until 2019. So what are they talking about here? And frankly, I don't know what they're talking about. And I don't know how this came about, but it sounds pretty weird to me. So if anybody has any more information, I'd love to hear it.